Hi, this is Muhammad. Welcome to my poster presentation. Plant viruses cross the cell walls through plasmodesmata
to spread between cells.
Therefore, plant viral genomes encode certain proteins to facilitate cell-to-cell movement. These movement proteins function by gating, dilating, and targeting the plasmodesmata. They are also movable between cells, and they have the nucleic acid binding ability. But this binding ability is sequence non-specific, so any movement protein can transport almost any viral RNA or any cellular RNA in their proximity. Many movement proteins can also complement movement of unrelated viruses. For example, the tobacco mosaic virus 30K movement protein can transport most of the tested viruses, including the PVX. That leads us to a question: how would then movement protein pick up their viral cargo? How they prefer the viral RNA over the cellular mRNA, especially at the early infection state where the viral RNA are not yet abundant in the cell. To answer this question, we have to understand the following studies. First, that the movement protein is the one viral protein that the movement protein is the one viral protein that doing the targeting to plasmodesmata and not the replication complex or any other viral protein. The second, mutation in any of the three movement proteins of the PVX, the TGBs, would render the virus movement incompetent and would not be able to move. And this is the key answer for the question that it was generally assumed that the virus moves as replicating. So, first the virus replicate before it is able to move. And this co-replicational movement hypothesis was reported recently by our lab when Tilsner found that 30K movement protein of the TMV and the 3 TGBs of the movement protein of the PVX were associated with the viral replication complexes near the entrance of the plasmodesmata, compartmentalizing the movement and the replication in a subcellular membranous structure near the plasmodesmata. These subcellular caps compartments provided protected environment for the virus from the silencing agents and concentrate the new viral RNA progenies into the PD channel, and that aids in the virus transport specificity. The question raised up again: how would the movement protein recruit this virus replication complex to the plasmodesmata? Two models may explain how. That could happen through a direct specific interaction between the movement and replication proteins, or indirectly via mutual binding to the same viral RNA or to the same host membranes or host proteins. It looks like the direct specific interaction is unlikely to happen, as one viral movement protein can transport many viral RNAs with different replication machineries. And to strengthen co-compartmentalizing, and to test this, we changed the movement protein. We swapped the TGBs of the PVX with the 30K, and we tracked the formation of the cap structures, and we examined these cap structures for the replicase and the viral RNA progenies, and we in fact found that using ecotopic GFP tagged replicase
using high efficient RNA binding protein,
we found that the replicase was localized in the cap structures and the naked viral RNA progenies were concentrated in them. And the 30K of the modified PVX virus was able to recruit the virus replication complex into the plasmodesmata, and as replication happens, new progenies were able to move. That would strengthen our co-replication movement hypothesis that the specificity of the virus transport
arise from mutual binding to the same factor, viral RNA or other host protein.